androgen receptor reveals differing conservation across varying domains. Did you know that prostate cancer is one of the most prominent non-skin-related cancers in the United States, affecting 1 in every 6 men in their lifetime? This cancer is commonly caused by the excess transcriptional activity of the androgen receptor. So what is the androgen receptor? The androgen receptor belongs to the family of steroid hormone nuclear receptors alongside the estrogen receptor and other receptors. The androgen receptor is a transcription factor that controls gene expression. Binding of the androgen receptor to its native ligands, 5-alpha dehydrotestosterone and testosterone, leads to male-specific developments, such as changes in physiology, reproduction, behavior, and in some cases, prostate cancer. The androgen receptor is a protein-coding gene and provides the instructions for the synthesis of the androgen receptor. Androgen receptors play an important role in male sexual development. The ligand testosterone binds to serum sex hormone globulin albumin to circulate around the body, and intracellularly, the testosterone is converted by 5-alpha reductase, an enzyme, into 5-alpha dehydrogentestosterone. DHT, 5-alpha dehydrogentestosterone, has a high affinity for binding to androgen receptor, and also binds important A to the androgen receptor in order to begin the translocation of the receptor ligand complex into the nucleus. Once inside the nucleus, these receptor ligand complexes dimerize and bind to androgen response elements, or AREs, in promoter regions of specific genes and help drive gene expression of these specific genes, contributing to the growth and survival of prostate cells. Androgen receptors also help regulate other important activities in both males and females, such as hair growth and sex drive. The androgen receptor gene itself is located on chromosome X, on Q11 to 12. The spatial arrangement of the AR gene consists of 7 introns and 8 exons, displayed on this figure here. The androgen receptor has 3 functional domains: the N-terminal domain, the DNA binding domain, and the ligand binding domain. A hinge region connects the DBD to the ligand binding domain. Androgen receptors are ligand-dependent nuclear transcription factors and belong to the steroid heme group of nuclear receptors, as previously stated. In this figure, we can see the chain of reactions that are created by binding to the androgen receptor. As we can see, after binding with after testosterone enters the cell and is converted into DHT by 5-alpha reductase, it is binded to the androgen receptor. These androgen receptors dimerize before entering the nucleus and begin affecting gene expression. However, there are also a multitude of other cofactors that can induce the androgen receptor, that can bind to the androgen receptor and induce it to dimerize and enter the nucleus, and also begin similarly affecting gene expression. So we know that testosterone and DHT is the main ligand that induces the androgen receptor to dimerize and affect gene expression, but there are a variety of other molecules that can also mimic this similar reaction. The N-terminal domain of the androgen receptor is encoded entirely by exon 1. And within this NTD, the length and sequence of the polyglutamine, CAG, and polyglycine, GGC repeats are variable in the human population. The length of these polyglutamine repeats have been shown to affect the folding and structure of the AR NTD. Removal of these polyQ repeats have been shown to lead to a reduction in alpha helical structure, whereas increasing the length of these polyQ repeats have been shown to result in a modest increase in alpha helical structure. The structural change on the NTD has an impact on protein-protein interactions, and the deletion of the entire polyglutamine sequence caused a 4-fold increase in AR activation compared to wild-type AR. Further deletion studies have determined the AF1 region as the primary effector region of the NTD. The AF1 contains 2 transcriptional activation units, namely tau1, located on amino acids 100 to 370, and tau5, amino acids 360 to 485. These 2 transcriptional activation units have been proven to be critical for full function of the androgen receptor. Additionally, the FQ-NLF proteins 23 to 27 and WTF proteins 433 to 437 motifs in tau1 and tau5 mediate ligand-dependent interactions between the NTD and the LBD that help regulate androgen-dependent genes and to stabilize the androgen dimer complex, which slows down the rate of ligand dissociation from the androgen receptor. So in this study, I will examine the phylogeny of the androgen receptor, specifically in primates, in order to gain a better understanding of the evolutionary history of the androgen receptor. Due to the importance and influence of the NTD region and the polyglutamine repeats, and the effect they have on human development, I hypothesized that there should be some moderate sequence variation in the overall NTD. However, in the DNA binding domain, there should be more critical conservation even among distantly related species due to the importance of the DNA binding domain. Results will be inferred by comparing nucleotide sequences and protein coding sequences across varying organisms via blast searches. So in order to conduct this study, androgen receptor mRNA nucleotide and protein sequences were obtained from the National Center for Biotechnology Information, otherwise known as NCBI. Nucleotide sequences were inputted into the NCBI's basic local alignment search tool, or BLAST for short, targeting the NTD, searching for common nucleotides. Subsequent BLAST jobs were performed on primates, applying further filters to search for specific species. 3 BLAST searches were required for the mRNA nucleotide sequence targeting nucleotides 1 to 2743, responsible for making up exon 1, and nucleotides 2473 to 3011, which comprised exon 2 and exon 3. 3 further BLAST jobs were performed, this time utilizing NCBI's protein BLAST tool to search for protein sequences instead of nucleotide sequences, again similar to the NTD and DBD region of human androgen receptors. Again, the BLAST tool was adjusted to search for 5 specific primate species, and only organisms of an e-value of 1 times 10 to the negative 4 or lower were used for comparison in the study to ensure accuracy. So these are the results I obtained from using the NCBI BLAST tool. As we can see, the 5 species chosen for the study were Homo sapiens, Pan troglodytes, Pongo abeli, Papilionubis, Macaca nemestria, and a lemur cata, otherwise known here by their common names. And here in this column we have the percent identity for the entire DNA sequence, and here we have the percent identity for exons, specifically for exon 1, and in this column we have the percent identity for exon 2 and exon 3. And here again, using the same 5 species, we have the percent identity for the entire protein sequence of the androgen receptor, and the percent identity for proteins 1 to 559, which encode for the NTD, and the percent identity for proteins 559 to 624, which encode for the DNA binding domains. So we can see that in the overall nucleotide, the entire nucleotide sequence, there is some relative variation, as well as in the protein sequence, there seems to be similar amounts of variation. However, in exon 1, nucleotides 1 to 2473, there seems to be a very mild amount of variation. However, in comparison to exon 2 and exon 3, where high amounts of conservation are displayed, there seems to be some nucleotide sequence variance in exon 1, which was what was expected. And in the protein sequence, similarly, there is some variance in the region compromising of the NTD. In this case, the proteins 1 to 559. And quite surprisingly, there seems to be 100% percent identity for proteins 559 to 624, which comprised exon 2 and 3, which is responsible for the DNA binding domain. So what do these results mean? Our data obtained through the nucleotide blast is consistent with this hypothesis. Across all primate species examined, the percent identity for exons 2 to 3 is greater than the percent identity in exon 1. For example, the Pongo bele has a DNA percent identity of 98.18 in exon 1 and a greater shared percent identity with the human androgen receptor of 99.63% in exons 2 to 3. And this sort of hypothesis is confirmed again in the protein sequence. However, it is interesting to note that although there is slight variation in the overall nucleotide sequence in exons 2 and exons 3 for 5 specific species, there seems to be 100% shared identity in the proteins, meaning every single protein was exactly the same. And this is likely due to the differing this is likely due to silent mutations which affect the overall nucleotide sequence but do not affect the subsequent proteins that are encoded from this DNA. So it's likely that there are small changes in this nucleotide sequence that although when examined display some variants when they are encoded into proteins, they are the exact same proteins are created from each sequence. Right. The NTD overall displayed a moderate level of variation. This is likely due to differing protein motifs which appear in the NTD, such as the trinucleotide CAG repeats, which have been shown to have an inverse relationship with the prevalence of prostate cancer, spinal bulbar muscular atrophy, and other conditions. And we can conclude that from our results, the NTD, which is responsible for transcription and may play a role in a host of diseases, is less conserved phylogenetically across species. However, more research is required to understand why and how these mutations occur in our evolutionary history. These are my references and notes. Thank you.